Hi, everyone. Thanks for joining us. Welcome back to Food Safety Office Hours. I'm Abby Snyder, Associate Professor of Microbial Food Safety at Cornell University. Today, we are talking about finished product testing. And so this is actually the last session in the short series in office hours that we've done in support of low moisture food safety. But yet again, this topic is relevant across many industries, so low moisture foods and beyond. Today, I'm joined by a panel of experts. We're going to start with an initial presentation from Professor Martin Wiedmann, Gellert Family Professor in Food Safety at Cornell University, and he will overview some of the opportunities and limitations associated with finished product testing for foodborne pathogens. That presentation is going to be followed by a discussion from Pam Wilger, microbiologist and fellow scientist at Post Consumer Brands. Pam has deep technical expertise in the methods used to detect foodborne pathogens from complex matrices, so she's going to give us a brief talk about that. And then after the presentations, we're going to open it up for Q&A. And I have a list of pre-submitted questions to work from, but as always, you are welcome to submit questions through the chat, or you can direct message me if you want to remain confidential at any time today. Joining us on that expert panel will be Dr. Catherine Carlin, Director of Microbiology and Innovation at Mariu NutriSciences, who also has technical expertise in product testing and low moisture food systems. And so with that, I'm going to turn it over to Martin for the first presentation.
I'm just going to pull up my slides. And so what I'm going to do today is just give a general sort of overview of some sampling considerations. And that's really focusing on the bigger picture. Take-home messages, and I think, it always is important to repeat this, even though most of you will be very aware of that. Finished product testing for microbial hazards can never assure 100% safe product. If you test 100%, if you want 100% safe product, you have to test everything, because finished product testing is what's known as destructive testing. You have nothing left to consume. So it is a tool, but it will not assure 100% safe product. This is a particular challenge if you were to test products of what I'm calling unknown provenance. You just get some dried product, some powder, you don't know anything about the facility, you don't know anything about the process, and you try to test to determine whether the product is safe. That's a losing proposition, essentially. But finished product testing for microbial hazards may have value in a few different scenarios, and specifically for verification, if you have a validated process, and if finished product testing is used along other verification methods, such as in-process testing, environmental monitoring, etc. Finished product testing, I think often when we talk about it, we think about pathogen testing, finished product, but there's also finished product testing for indicators, such as standard plate counts, aerobic plate count, Enterobacteriaceae, and other indicators, which can be valuable as a tool for process control. Essentially telling you, hey, do I have a consistency in my process, or is there a lot of variability? And you can do that to make sure your process is in control to make a safe, sufficiently safe finished product. You can also use potentially finished product testing for process validation, but that's really a different topic where you would have to inoculate product potentially to do this, or have a really good surrogate, which is not trivial. I think there's sometimes confusion about this, and I'm going to take the example of milk pasteurization. I think, we all sort of assume, and correctly so, that pasteurization of milk gives us a 5-log reduction of target vegetative pathogens. And people often get very concerned about this if they actually just do a standard plate count of raw milk before and after pasteurization, and they only see something like a 2-log reduction in total bacterial numbers. So what you see in total bacterial number, naturally occurring bacterial number reduction, doesn't necessarily reflect the log reduction you get from your pathogen control process. So that's important to consider when you do process validation. Now, when you do finished product testing, there's many dimensions you need to consider in order to determine whether you're going to do it, how frequently you do it, how many samples you collect. And that includes everything from regulatory compliance to risk tolerance and trade-offs. And I'm going to get a little bit more into some of these details. So food production requires a validated process to make sufficiently safe food that then is consistently verified. I always like to just make sure we're on the same page with what we mean with validation and verification. Validation is, are you doing the right thing? Do you have a process that assures a sufficiently safe product? Verification then is making sure that you consistently implement that validated process every day, every minute of your production, essentially. And the textbook for this is obviously pasteurization, right? I mean, we have, and we use it in the dairy industry, but it's similar in the meat industry, many other industries, and it goes beyond pasteurization. It goes to essentially all validated heat treatments. We typically have scientific data that give us a specific temperature and a specific time. For pasteurization of dairy product, that will be 72 degrees C for 15 seconds, assuming it doesn't have a higher fat content. And we know, and the scientific data are there, and really essentially undisputed, that this provides more than a 5-log reduction of the vegetative pathogens of concern. Right? That's our validation. Our verification then will be continuous recording of temperature and time to assure that every drop of milk in this example meets those, it goes on through these processing conditions. Okay. So let's try to do a reality check on finished product testing. And I think to illustrate some of the challenges. Let's say you have a hypothetical raw material that has 1 salmonella per 100 grams. Could be in some products, and it might be higher, but it doesn't really matter. The math will be similar. You have a heat treatment process that gives you a 5-log reduction, so you get substantial reduction of those salmonella to produce a food that is essentially agreed upon is sufficiently safe for commerce. Now, a hypothetical company now on a given day, one day, makes a product, but the heat treatment only delivers a 2-log reduction. And that can happen for different reasons. It could be our pasteurization wasn't applied properly, but our verification data would have caught that. It could be something more complex that if we have to dissolve, let's say, fibrous material, and we have a certain dwell time, we didn't follow the dwell time, and now salmonella is heat resistance, that wouldn't be as easily caught, etc. Now, we do finished product testing. Now, if you have a 2-log reduction from 1 salmonella per 100 grams, you now have 1 salmonella per 10 kilograms, because 1 salmonella per 100 grams is 10 per kilogram, so 100 per kilogram. A 2-log reduction of 100 per 10 kilogram is 1 per 10 kilogram. So, not really sufficiently safe yet. How likely is a finished product test picking up 1 salmonella per 10 kilograms? Okay. I am not aware of anyone that really would test 10 kilograms of finished product for salmonella. So even with a failure that's quite substantial, okay, it's a relatively low raw material and a substantial 3-log rest reduction that you should have, finished product testing isn't going to pick up that problem. To just sort of set the stage before we get into, or instead of getting into higher math, is what the complexity is of this. So, but this should not happen because we will have our verification. We have our verification of a time temperature. We have our verification of dwell time. We have all these other parameters in terms of a standardized process that assures the safety of the product. Now, there's tremendous allure of finished product testing. And it typically sort of shows itself by a customer, let's say a retailer saying, hey, we need you to do finished product on the product you sell us. And and it's because obviously, and typically the logic is, well, we know doesn't assure a safe product, but we just heard of someone that did finished product testing and it found a positive. And the fact that they found a positive for a pathogen proves that finished product testing is valuable, which is not true, okay? But why is it not true? I'm going to go through another hypothetical. Assume you have a large facility, you make a bunch of lots of any product you want to imagine that has very low salmonella load, 1 salmonella per 10,000 kilograms, 20,000 pounds. You can pick whatever number you want, okay? For simplicity of math, let's assume you test 1 kilogram of your finished product. Most people don't do that. They do it at 750, 1,500, but I want to keep the math simple, okay? So that means if you have 1 salmonella per 10,000 kilogram, your chance of finding a positive is 1 to 10,000. But it's like the lottery, right? I can test today and have a positive just because, let's say, I was unlucky or the lightning struck or whatever analogy you want to use. But that doesn't mean that product tests positive is any less safe than the 9,999 other products that also had 1 salmonella per 10,000 kilograms, but didn't test positive. It was just the luck of the draw, essentially. So that argument that someone found a positive and it proves the value of finished product testing to assure safety is absolutely substantially flawed. And this is very, very important to keep in mind as we have these discussions. Now, finished product testing additionally can have some substantial trade-offs. And this is modeled on a conversation I had, in the fairly recent history, where we had a company that did a lot of finished product testing and produced a lot of product. And so let's assume, for simplicity's sake, they produce 20 lots a day, and they test each lot for pathogen X. And let's take a ridiculous example. Let's say it's Vibrio. Okay. Now, each test costs $25. So this company were to spend $500 on testing for Vibrio, which isn't going to really be a hazard in their product. That means if they run 300 days a year, it's $150,000 in terms of total cost of testing. Now, as a food safety professional, we need to ask yourself, hey, do I have a better use or have a bigger food safety impact with $150,000 than doing finished product testing of a pathogen that I don't expect there? And if it's there at low levels, it probably doesn't cause any issue. And where I have a number of other controls where I can do some very cheap direct monitoring sort of time of temperature, isn't that enough? And for example, my example is that, hey, it's $150,000. I could hire 2 more people on sanitation. Isn't that going to have a bigger impact than finished product testing? So the argument that, well, we got to do the right thing, we got to do more finished product testing, always assumes that there's unlimited amount of money, and it ignores the concept of opportunity cost. Is there an opportunity to spend that money that one might spend on finished product testing on something else that has a bigger impact on finished product, on the safety of the product? Okay. So I gave you some of the ideas of what the problem is with finished product testing. And I'm not saying you shouldn't do it. I think you need to understand the limitations. You need to then pick it very carefully and understand what is your goal. Is it verification? And what else do you have for verification? Because even for verification, finished product testing alone would not be sufficient. You need to consider, back to the bottom of my take-home message, what are you trying to achieve? Are you trying to achieve regulatory compliance? What is your risk tolerance? Are you really reducing your risk with it? And what are the trade-offs associated with finished product testing? So now let's dig a little bit on what I would call sort of take-home messages that some of you might be actually practical. Use, if you want to do finished product testing, if you are doing finished product testing, make sure you understand that it has to be part of an overall strategy to assure the safety of your product. You cannot and should not, in my mind, plan or assess finished product testing in isolation. It has to be in the context of your other controls, your other verification activities. You need to consider the benefits as well as the trade-offs of finished product testing. Remember the concept of opportunity cost. Define the goal of your finished product testing. Okay. Is it compliance? Is it regulatory compliance? Or are you trying to just comply with customer requirements? Is it verification? Is it process control? Is there some other reason why you're doing it that I could not imagine or come up with? Decide on what your target organism is. Should you test for pathogen? Should you test for indicator organism? Should you test for both? And then the important thing is when you do finish product testing, you need to always have a very clear plan for a follow-up on positives. For example, is root cause analysis. That's where the real, impact on food safety happen. It does not happen by, not sending that one lot that tests positive for pathogen not into commerce. It happens by root cause analysis and controlling that hazard introduction, closing that loop that made that product be positive. However, be back to the example, you may not always find a root cause because it may have just been the luck of the draw that you have a very, very low contamination of your finished product, and you may have just had one positive. And there's been some studies out there that tried to do follow-up where you had a positive and then do additional testing. And sometimes we find that there are additional positives that identify some sort of more systemic error and issue. And sometimes we see that it almost doesn't matter how many follow-up samples, additional samples we take, we're not going to find another positive because it is really that random chance finding that lightning strike that just happened. That's not indicative of anything larger. We need to keep that possibility in mind when we do this. So those are my thoughts. Looking forward to what Pam has to say and looking forward to the discussion. Thank you.
Audio. All right. Thanks, Martin. I'm going to share my screen. And hello, everybody. Thank you for attending. One more click. Or two. Everybody can see it now? Okay. I just want to get rid of it. All right, so I'm going to talk then about the actual testing method for low moisture matrices. I do not have this silver bullet or this big aha moment, but what I hope you walk away with are some questions to be asking your testing laboratories or your leadership you work with. We should also, hopefully you will take away some things to think about as well when you are picking out your methods. So low moisture products, way different than what I consider wet products. So I call them dry, the dry industry versus the wet industry. And to me, the wet industry is a lot of the animal protein, a lot of dairy, things like that. But low moisture products are a huge part of the food industry, and things that we don't necessarily think about as being low moisture always come to mind would be things like peanut butter, chocolate, even like some of our syrups, things like that, but it's all defined by your water activity. Huge difference now is that when we tend to make these low moisture products, we are making them continuously for many days. I'm talking anywhere from 2 weeks to 45 days without stopping. And so what's going on there is if you have any kind of water induction into your system or contamination that can have water, because we have to induce water for the organisms to grow. Otherwise, running continuously at a hot temperature or a certain pressure or flow, we tend not to have microorganisms growing. And so that's the benefit for us as dry producers. We don't have that growth. But just to let you know too, you need to have a study that actually will validate your run length. So if you choose to run for 45 days, 60 days, people like to see and they want to understand that for microbiology, the microorganism counts are not increasing over that time. And that's what we typically see. So again, we would have to run a study with that. And a lot of times we just do total plate count, total Enterobacteriaceae. This is not a pathogenic exercise. The other thing to think about is if you have low moisture products, it's not homogeneous as like liquids. It's a lot easier to keep liquids homogeneous, but our dry products, it's not going to be. And part of that problem too is we are dealing with a very low level of contamination because like I was just talking about, without that added water, without a higher water activity, you're not going to get growth of any of these organisms. So again, your contamination is usually somebody like cross-contaminated it, either through touching, maybe I didn't clean or sanitize my equipment as good as I should have, or worse yet, your so-called validated kill step isn't working continuously or consistently, and you're getting a different variance in the amount of contamination you see in there. Another area that you want to keep in mind is it's just like what Martin said, that we use a lot of indicator testing to just show that we are making our products consistently. Our process is consistent. The only way to show that microbiologically is by testing these microorganisms. You can, of course, you should be monitoring times and temperatures, pressures, and things like that, monitoring your process. But in the end, people are always asking about, but what are the microorganism counts telling us? And this is critical, especially after you do a wet cleaning and sanitizing. You might run ATP to test cleaning, but that does not measure and give you a microorganism count at the low levels we are targeting. So it could tell you if you have a high number of microorganisms, but a visual look at your equipment should tell you that as well. So you don't use ATP to measure microorganism level. You're going to have to literally take a microorganism swab. And so what we tend to do is we look for Enterobacteriaceae, and these are going to be on zone 1 pieces of equipment. And we are always targeting less than 10. Some people also do total plate count, and in order to do total plate count on these zone 1 surfaces, you need to set a baseline. We do go for a target sometimes of less than 100 if you're really good, but you might have to go up to less than 100. So it is, again, something you're going to have to test and understand and then set that target. And this is really where we do a lot of these enumeration tests. Anything special for enumeration testing? Really, the part is a lot of those are swabs, nothing different than what I do swabbing in a wet environment. So methodology, no, we don't have to check with that. Just have to make sure that they're validated for surface swabs. If I was to do an enumeration test, though, for the product, you have to think about things, especially powders, that when the person is combining the sample with that diluent, whatever the diluent is, they might have to pour that sample very slowly so they don't form clumps, because then you're missing half of your sample to actually touch the diluent, and then you're going to get an inaccurate result. So sample preparation is critical when it comes to low moisture products and enumeration tests. The last thing I want you to keep in mind about low moisture products is because it is a low level of contamination typically, we are running continuously in our facilities. Time to that final result, which oftentimes includes confirmation, is really critical to keep that to a minimum, because you are running your facility that could be making hundreds of thousands of pounds after your presumptive. And how do I know that presumptive is going to end up confirming or not confirming? Do I shut down my facility when I get a presumptive, or do I continue running? And there are some tests out there, such as the BACS real-time PCR. They have validated a secondary step after a presumptive test that you would run 4 quads. Well, we call it the quad testing. So you go back to the lysis and you will run 4 more tests on the PCR machine. And if all of those are negative, that original presumptive is called negative. If any of those are presumptive positive, you need to move forward with your cultural confirmation or whatever confirmation method you have deemed necessary. This has saved us a tremendous amount of time because, as we all know, or maybe you don't know, but there are no perfect methods out there. And it's not that the method is inaccurate or like failed type. I don't like to think about it like that. But because there's no perfect methods, at times you'll get something that's considered atypical, and those tend to go negative, but the result says it's presumptive. So a lot of decisions are being made off of this, and we really want to then get to a point where when we do detect this organism, we know that it's truly accurate and it's truly going to be positive. So another thing to think about is, and Martin touched sort of on this he talked a lot about other kinds of validation, but you also have to do method validation. And that, just like in process validation, is different than method verification. We really need to show that the methods that we are choosing are fit for purpose. So a method validation, things that you have to keep in mind when you have a method validation is, what was the sample size I used? So we all know for Salmonella, we typically use 375 gram samples. So you would validate that method at 375 gram samples. But let's say one day I want to just test 25 grams. So if I validated the method for 375, that method is considered validated for all the smaller samples. So it would be validated for 25 grams. However, if I validate a method for 25 grams, and then tomorrow I want to test 375 grams, it is not validated for 375 grams. So you would have to have another validation study on that method. Now, method validation for low moisture products is where we get a lot of problems. These are considered challenging matrices, and on the next slide, I'll talk about those, and can be very difficult. So things to watch out. I have found that the best way to have method validation done. Now, first and foremost, I must say this. All the methods that are available today commercially have already been through a method validation scenario. They have AOAC approval, AFNOR approval, microval approval. They have gone through that. The problem is, in order to get that validation, they only had to test so many different matrices, so many food categories. Typically, it's 5 food categories if you want to do a broad claim, and it ends up being about 20 different product types. The problem with that is we have probably more like 20 million product types. And so the validation may not work for your specific matrix. And that's where you could also then run a method verification and showing the methods fit for purpose. And that also at times could be called a matrix extension. This is very overwhelming if you're not into this kind of part of the testing. So I would suggest the best thing for you is talk to whoever is the manufacturer of your test method and have them help you through this validation. Maybe they already validated it with the sample size and they can explain that to you and show you so you're confident. Or if it hasn't been, that you could then do method verification with them. So we want to, again, make sure you can show the method you're using is fit for purpose. And that includes sample size. The other thing we want to talk, should I be testing a composite sample versus a grab sample? So when the Salmonella sampling plan was written by the FDA many years ago, it talked about grabbing 15 100-gram samples throughout your production lot and then taking 25 grams from each of those 100-gram samples and combining them into a composite sample to make 375 grams. Now, most people are just grabbing the 25-gram samples. They don't even do the 100-gram samples, but they're doing grab samples. And there's studies, and it's published studies showing that if you grab smaller amounts of product more often, that is going to give you the best chance to find this one cell of Salmonella in that 22 pounds or the 10,000 pounds that Martin was talking about. Because again, we are trying to keep our consumers safe, the public safe. So we want to find that one cell. And in order to do that, it starts with the sampling. So how am I going to grab small amounts often throughout my lot? And that typically brings you to an auto sampler. Now, auto samplers are not perfect either. One, you have to make sure you keep those clean. You don't want to contaminate your sample from your auto sampler. And two, where am I placing the auto sampler? Where can I put the auto sampler? Ideally, you would want to put it as close to the end of where you're sampling your product or packaging your product as you would when you grab your samples for testing. So keep in mind, the more samples I can grab over time throughout production, the better representation of finding something in our low moisture products. Then there goes question of sample frequency. That is going to be driven about how you defined it. Martin talked about you need to define what am I trying to answer with this test method. So that should drive the sample frequency. So when I talked about grabbing individual grab samples now to test the sanitation, cleaning and sanitation program, I want to grab ideally the first product coming off my line and then another grab sample every hour after that for, let's say, 12 hours. Because if I didn't do a good job cleaning and sanitizing, I also then will see this through these finished product enumeration tests, because you'll see higher counts and you would see it going down over time. And so that is another way to verify how is my cleaning and sanitation program working. Then two, like we also, so like pet food, peanut butter, we do things called rolling lots because it is and there's probably other products. We make it continuously. We define what we call the microbiological lot, which is how much product are you going to make? And from that amount of product, we could have 2 hours of product, 1 hour, you could have 8 hours, you could even define 24 hours. And out of there, how many samples am I going to grab to make either the 375, 750, 1500 is what you define. So again, you really need to understand, what are you hunting for? How high is it? How high do we expect it to be? And then how much am I making? And Martin and I were thinking a lot alike on this, is you can't test safety into the product. So totally agree with this. People feel that if their supplier gives them a C of A that says salmonella negative or absent, not detected, that they're safe, the product is good. That result is only for the sample that they're reporting. It means nothing else. So what you have to do is you really need to understand what that supplier is doing to prevent, first and foremost, contamination getting into their product, and how to not recontaminate that product. So I do not really care. It's a strong word. It doesn't mean a lot to have a C of A with a salmonella negative. I have yet to see anybody send me a product with a C of A that says salmonella positive. So you could just understand and know that if they're shipping your product, they probably feel that it is salmonella negative. Okay, so some matrix challenges. Peanut butter and chocolate. I already mentioned those and how we test it. But the problem with peanut butter and chocolate is the fat content. And we all know that fats and waters don't really mix. So when I am putting my product into that liquid medium, which is typically made from water, could mix it. I can mix it and try that, but then it's going to incubate for, let's say, 24 hours. For after maybe 5 minutes, you're going to get the majority of the product to lay on top of the liquid. This is true for the deodorized, bleached, refined oils. How do I get oils, fats, and water to mix so that I know that I can pull the contaminant out of that layer and have it grow in the liquid medium? So you need to be thinking about this, and maybe it has to do with your sample size. Maybe you can't use as big of a sample size. You have to go smaller and check it more frequently. But that is a major problem with the peanut butter and chocolate. We were validating our cereal with our method, and we picked what we thought were higher risk cereals, which could be cereals that have things added after the kill step, such as almonds, freeze-dried fruits, raisins, things like that. But then the interesting thing we found is that our cereal that has cinnamon in it did not validate. All the other cereal validated, but the cinnamon didn't. And all it meant is our method had an extra step that was needed to be done. And as long as we added the extra step, the cereal with cinnamon was then a validated method. That was really surprising to me. And again, we should have thought about that, because we know that we struggle with spices and how challenging of matrices they are when it comes to testing. But even that little bit of cinnamon in our cereal was also causing us problems. Salt, I worked in that industry, and I've had lots of people ask me to do micro testing on salt. So can you imagine adding 375 grams of salt to 1 to 9 dilution? No methods are validated for that much salt. You're really going to struggle with that. And then we also have had lots of questions. I've worked in the refined oils industry. Lots of people ask and are testing these products. There's no validated methods for oils either. We just can't figure out how to make it to work. So you really need to question why you're putting so much energy on these products that are very low risk and how they are processed, as to why you really feel you need a microbiological test. There's no methods available. Probiotics is another one, whether it's a concentrated probiotics or it might be pet food that has probiotics added to it. How much are the probiotics going to interfere with me finding salmonella? Another thing to make sure that your method developers or your testing lab knows what you're dealing with so that they can define the validation study appropriately. And with that, I'd just like to thank you, and I think we're going on to questions next.
That is right. Thank you
All right.
both to Pam and Martin for your presentations.